This animation depicts the hypothesized process of evolving the anti-CRISPR protein AcrIIA4 to inhibit Pyogenes DX-Cas9, utilizing our iGEM team's novel Q-beta directed evolution technique in vivo. Directed evolution of AcrIIA4 to inhibit Pyogenes DX-Cas9 in vivo. As an introduction, there are 3 vectors used in this process. The first is comprised of the chloramphenicol resistance gene, CMR, and the superfold green fluorescent protein, SFGFP. The CMR is constitutively expressed, allowing the plasmid to defend itself from the selective pressure of the chloramphenicol, CAM, present in the LB media. By pairing expression of the SFGFP to the same constitutive promoter following CMR, fluorescence can visually represent the quantity of cells that have survived the pressure. The second vector is made up of the Pyogenes DX-Cas9 protein and single guide RNA, sgRNA. The 20 base pair variable region of the sgRNA leads the DX-Cas9 protein to bind to the CMR gene, hence blocking the expression of both chloramphenicol resistance and, by extension, GFP fluorescence. Finally, the third plasmid contains the anti-CRISPR, AcrIIA4, which has the potential to inhibit DX-Cas9 when flanked by 2 MDV promoters. Successful inhibition of the Pyogenes DX-Cas9 by the AcrIIA4 protein would unblock gene expression and thus allow the cells to survive the selective pressure of the CAM-LB media. These 3 plasmids work together, as demonstrated in the following slides, to create selective pressure to produce AcrIIA4 that is capable of blocking the DX-Cas9. To begin, E. coli with their 3 plasmid system are grown in the LB media with chloramphenicol. With the first plasmid, we have our CMR and GFP constitutively expressed. However, when proteins from the second plasmid are synthesized, the sgRNA guides the Pyogenes DX-Cas9 to effectively bind to the CMR gene, blocking expression of both resistance and fluorescence. Initially, non-mutagenized AcrIIA4 protein is produced that is not evolved to effectively knock DX-Cas9 off of the DNA, and thus the CMR is not produced. Consequently, this would cause the cell to be vulnerable to the antimicrobial presence in the media, leading to its death. However, now let's take a look at what happens over time given the MDVs, also known as Q-beta binding sites, that flank the AcrIIA4 gene and thus enable Q-beta directed evolution to take hold. The later generations of AcrIIA4 undergo mutagenesis, leading to equivalent, weaker, or possibly stronger forms that are less or more able to bind and inhibit DX-Cas9. Due to the combination of genetic drift and the selective pressure of slowly increasing chloramphenicol, the leading mutated AcrIIA4 from the latest generation will be chosen and integrated into new LB media with an increase of CAM concentration. Once again, some descendants will have mutations. The strongest proteins, those which survive the selective pressure, will be passed on generationally when the chloramphenicol dosage is increased further. This newly mutated anti-CRISPR will then effectively inhibit the Pyogenes DX-Cas9 protein, allowing for the return of chloramphenicol resistance, which would cause the cells to live with the selective pressure of the media. Although we are unable to accomplish this portion of the project due to time, we wish to do so in the coming years.